weakened is by the muscle. Now, what is the first starting point? Which channel have to open? Sodium channel have to open. The first starting point is sodium channel opening. Sodium comes inside, leading to depolarization, which further opens this important group of channels, calcium channels. First sodium comes, then calcium comes. This calcium is very, very important to push the acetylcholine containing vesicles outside. What we call this important process as exocytosis. For exocytosis, you need calcium. Which neurotransmitter is going to get released? Acetylcholine. This acetylcholine have to go and act in the postsynaptic muscle end plate. Which receptor? Acetylcholine. Very important. Disease is also there. Nicotinic acetylcholine receptor. So acetylcholine acting in this nicotinic acetylcholine receptor in the muscle end plate now. It's a special type of sodium channel. Sodium ions coming inside, leading to a special type of potential that is called end plate potential. The sequence they asked recently in INACET. First sodium comes, then calcium comes. Exocytosis of acetylcholine. Acetylcholine acting in nicotinic acetylcholine receptor, leading to release of sodium into muscle end plate, leading to end plate potential. This end plate potential further develops into depolarization, which opens certain calcium channels in the muscle, leading to release of calcium. This calcium is very, very, very critical for muscle contraction. So inside the muscle now we are seeing this sodium influx leading to end plate potential. If it crosses threshold, we will get depolarization leading to opening of calcium channels. This calcium channel is the one which is going to cause muscle contraction. Now the big question, what are these calcium channels found in the muscle? Any idea? Calcium channels found in skeletal muscle, three important types. First and second I am showing here. Both these two are very, very, very critical for contraction. Who are all they? Dihydropyridine receptor, ryanodine receptor, muscle calcium channel. What type of interaction they both are having? They are having, remember, INICET MCQ, a mechanical type of interaction. This channel configuration changes during muscle contraction. Ryanodine receptor releases. It is a release channel, release calcium, and this calcium is very, very critical for contraction. By any chance, if this ryanodine receptor is overactive, ryanodine receptor, you get a very fatal condition. What is that clinical condition, sir? Remember, guys, malignant hyperthermia. Their normal activity, release of calcium, muscle contraction. If they are overactive, enhanced release of calcium, leading to a fatal disorder, malignant hyperthermia. So dihydropyridine receptor, ryanodine receptor, both are for contraction. Following contraction, eventually relaxation will take over. For that, the third muscle calcium channel. This I told you already. What is this channel protein? SEPCA. Underline this. Yes, it is concerned with the storage of calcium, not release. Release means contraction. Storage, all the calcium is taken back and stored. It eventually lead to muscle relaxation. So dihydropyridine, ryanodine receptor for contraction, release of calcium. Storage of calcium, SEPCA pump, no more calcium available, all are stored back, muscle will relax. See, this neuromuscular junction we are discussing in detail because of a critical importance. This transmission can get blocked. Where all it can get blocked? This voltage gated sodium channel is blocked by a very important toxin, tetrodotoxin. Okay, you usually get this from puffer fish. Tetrodotoxin. Next, there is a potassium channel which can be blocked by dendrotoxin. Potassium channel blocker. The calcium channel we have discussed. It is blocked by phonotoxin. At times they turn fatal. Tetrodotoxin, sodium channel blocker. Dendrotoxin, potassium channel blocker. Phonotoxin, calcium channel blocker. And one very, very, very critical. Remember this acetylcholine. Block release of acetylcholine. Who is that?
botulinum.
Toxin, remember, botulinum toxin, microbiology. There is no acetylcholine release. That means no muscle contraction. You get a classical type of flaccid paralyzers. Very important. So which toxin is going to block? Which toxin is going to block release of acetylcholine? Botulinum toxin. Heterotoxin, dendrotoxin, conotoxin, botulinum toxin. What about diseases? Any diseases there? Neuromuscular transmission? Yes. If there is development of autoantibodies against this calcium channel, if these calcium channels are destroyed, you get a very important clinical disorder, that is Mbbs. What is this, sir? This is our
Lambert-Eaton myasthenia syndrome.
Myasthenia.
It is due to
Very important.
calcium channels, while myasthenia gravis is due to nicotinic acetylcholine receptors.
Calcium channels.
Nice.
Lambert-Eaton myasthenia syndrome. On the other hand, even more important, the same autoantibodies, if they destroy this acetylcholine receptor, nicotinic acetylcholine receptor, we call this a very familiar disorder, that is our myasthenia gravis. Autoantibodies against nicotinic acetylcholine receptor, myasthenia gravis, muscle weakness. Autoantibodies against voltage-gated calcium channel, Lambert-Eaton myasthenia syndrome. Alright guys, now we are going to understand the molecular mechanism of muscle contraction. That means, effectively we are going to discuss about a very important theory. What is that theory, sir? Our very well known sliding filament theory. To understand this, we have to first remember there are 4 steps. 1, 2, 3 and 4. Next important point. This is going to be our actin. This is going to be our myosin. And this particular part is the head of myosin. All the process start, the beginning point is, remember guys, ATP coming and binding to myosin head. Whenever ATP binds to this myosin head, visible observation, its head is oriented at 45-degree angles. Immediately on ATP binding to myosin head, an important enzyme will be activated. What is the enzyme, sir? ATPase. What does ATPase do? It will split this ATP into ADP and a phosphate. Whenever ATPase is activated, it hydrolyzes our ATP to ADP and the phosphate. Most important part. Because of this hydrolysis, we are clearly seeing the head now shifts to 90 degree. Because of hydrolysis of ATP, from 45-degree orientation, it now moves into 90 degree. Now in this head, what is attached? ADP and phosphate. Because ATP is hydrolyzed, what we are having is ADP and phosphate, and the head moved to 90 degree. This is step 2. Now further process will continue only in the presence of calcium. You need calcium to move into step 3. What are we seeing? We are clearly seeing this myosin head at 90-degree orientation, still having ADP and the phosphate, goes near an actin molecule and combines with it. What we call this important step, remember guys, this is our classical cross bridge formation. In simple way, actin myosin interaction. It is not happening here. It happens only in the presence of calcium. What is happening? Cross bridge formation. So step 3, cross bridge formation, 90-degree orientation, ADP and PI. It's a transient attachment. Quickly this ADP and the phosphate will be released. Because of this release, the head now moved from 90-degree to 45-degree. In that process, this myosin is now overlapping or sliding to the next actin. Previously it was here. Now because of this ADP and the phosphate release, it is now slided. What is this happening here? Sliding. What is the other name for this sliding, sir? This is our power stroke. See, ADP and the phosphate is released. This 90-degree head will now convert to 45-degree. In that process, moves to next actin. 45-degree moves to next actin. 45-degree moves to next actin. What is the sliding filament? What is also called? Power stroke. So step 1, step 2, step 3, step 4. How the cycle will continue back again? Look very carefully. Now this head is free. There is no ADP and phosphate. Most important point. For myosin head detachment, and to start a new cycle, you need a molecule of ATP. This is the area they target always in INICET. For the cycle to continue back again, for this myosin head detachment, see it is detached here. For that, you need a new molecule of ATP. When ATP comes and bind here, the entire cycle is repeated. It keeps on continuous as long as ATP is available. So myosin head detachment requires a new molecule of ATP for the cycle to start again. This is our sliding filament theory. Now we are going to understand a few properties of skeletal muscle. The first one is obviously about the relationship between length and tension.
Guys, this is the Neet PG 2025 question. This is,
There are 4 points.
I made it wrong because I haven't studied this physiology.
Always remember guys, this tension generated is always directly proportional to actin myosin overlap. So optimal overlap means more tension. Not much of an overlap means less tension. Now let's look into point D. We are clearly seeing at the point D, the tension is minimum. Point D, the tension is minimum. Why is that? See guys, this is our myosin. This is our actin. overlap perfect. They are optimal. How is this? It is true? Yes, there is optimal overlap between actin and myosin. On the other hand, at A, the tension decreases. At point A, the tension falls. Why, sir? Overlap is still there, but this overlap is not optimal. What I mean? Between B and C, there is optimal overlap. Very good. But at point A, there is overlap still there, but it is not optimal. Tension is there, but it is decreasing. So point D, minimum tension. Point B and C, maximum tension. Point A, tension decreases. If they are widely apart, no overlap, no tension. If there is optimal overlap, tension generated will be good. That is why at point B and C, there is optimal overlap, tension generated is maximum. Simple. Now, inside skeletal muscle, if you see certain fiber types, look here. This is first type, my dear friends. It is usually what color? Is it big or small? It is small. Type 1 muscle fibers. What about the next one? You have type 2. Those type 2 fibers, visibly they are large. Clear? Type 1 is small. Type 2 fibers are large. In this you have 2A and 2B. This large 2A is also red in color. But this 2B, we are clearly seeing it is white in color. So how many type of fibers are there? 1 and 2. 1 is small and red. 2, there are 2 subtypes, 2A and 2B.
इधर मस्त बात ये है कि 1, 2, 2A and 2B ये हमने पढ़ा था now में भी, but it's not now, it's muscle.
They are large. 2 is large. 2A is red in color, but 2B is white in color. Why they are red in color? What is the reason? What functional significance does it have? What is the meaning of white? What is there? What is absent there in white fibers? We are going to understand. Right, guys. Let's understand the significance of this muscle fibers. See, we have type 1 fibers. In type 2, we have 2A and 2B. We'll make this table look very simple for you. Let's start from what we have known already. See, type 1 fibers are red in color. 2A we have seen red in color. 2B, it is white. Second, the diameter of type 1 is small, I have shown you. 2 fibers in general are large. Now, even more minute points to look into. Why red in color? Blood is red in color because of hemoglobin. Muscle is red in color because of myoglobin. Red means myoglobin present. Red means myoglobin present. White means no myoglobin. Most important point. No myoglobin means no oxygen storage. They predominantly depend only on glycolysis. It is for this reason. Type 2B fibers, we know they are white. Type 2B fibers, we know they are large, but they predominantly depend on glycolytic mechanism. So 2B is white, no myoglobin, no oxygen storage. They predominantly depend upon glycolysis. They are large. Now, let's fill the box for type 1 fibers. We know they are small. We know they are red because of myoglobin. Oxygen storage is possible. That means they can undergo oxidative phosphorylation. So oxidation is possible. Glycolysis, anaerobic glycolysis is possible in 2B. Oxidation is possible in 1. We are strategically placing this 2A in between 1 and 2B. Why? Now let's fill the box. 2A is large. Because of myoglobin, it can undergo oxidative phosphorylation. It is large. It can also undergo glycolysis. Both are possible. Very simple. Type 1 is small, red, oxidative phosphorylation. Type 2B is white, large, glycolysis. 2A is in between these two. Large, oxidative, glycolysis. Both are possible. Now, very important. This we have seen, small, large, large. Glycolytic capacity is very, very, very high for 2B. We have seen. The glycolytic capacity is very less or moderate, we call for 1 fibers. Next, recruitment order. Usually, 1 fibers are recruited first. 2A is recruited second, and 2B is recruited last. Why? What is the simple understanding? Say I'm sitting, guys. If you ask me to run, from sitting, I have to move into standing, then I have to run. That's all. For sitting, body uses more of 1 fibers. For standing, during standing, my body will recruit 2A. If I have to run, the running part comes last. This last running part, 2B. That's all. Remember the other names, everything is simple. 1, small, red, oxidative. 2B,
1 small
large,
red
white,
oxidative.
glycolytic.
2B
2A is
large
actually in between these two.
glycolytic.
2A is large, it is red, it uses both mechanisms. Both oxidative phosphorylation because we have got myoglobin. You can also use glycolysis. Usually for all body functions, recruitment, 1 is recruited first, 2A is second, and 2B is last. Activity best suited. This type 1 fibers are for sitting, recruited. Type 2A is recruited during standing, and 2B last during running. That's all. This skeletal muscle, if you continuously stimulate, see we are continuously stimulating.
Tetanizing,
yes,
tetanizing.
That means only contraction.
Sustained
There is no relaxation.
contraction.
Remember, a
No
state of
relaxation.
sustained contraction without any relaxation, we call this as tetanic contraction. Continuous stimulation.
Cardiac muscle
State
don't
of
have a
sustained
tetanic
no relaxation.
contraction,
We
if
call
you
this
remember.
tetanic contraction. How to find out this frequency, sir? If you stimulate in this frequency, you will get tetanic. That is why we call this as tetanizing frequency. In exam question, they will give you the contraction period. Tetanizing frequency we calculate by 1 by contraction period.
1 by contraction period.
Tetanizing frequency equals the inverse of contraction period, 1 by contraction period. A state of sustained contraction without any relaxation, we call this as tetanic contraction, tetanizing frequency. Now, from skeletal muscle, let's move into cardiac muscle. Our skeletal muscle is under our voluntary control. You can command it to contract, but cardiac muscle, you cannot command it to contract. It is involuntary. Most important first point. Cardiac muscle is striated, but it is involuntary. Next important point. You see extensive amount of gap junctions. In heart, they are located in intercalated this region. What protein you will see here? A very important gap junction forming protein, connection. Because of this connecting gap junctions, all cardiac muscle exclusively contract in synchrony. What is the synchronizing contraction called as functional syncytium. So through gap junctions, the impulse passes to all the cardiac muscle cells. They contract together at the same time. This concept is functional syncytium. Exclusively in cardiac muscle, you see a phenomenon calcium-induced calcium release, which means you first need calcium from the extracellular source. This extracellular calcium is needed to release this later calcium.
Cardiac gap junction and connecting.
So extracellular calcium is needed, very important, exclusively in cardiac muscle.
Extracellular
For release of calcium from the
source.
intracellular sources, we call this as calcium-induced calcium release. One other protein you see exclusively in cardiac muscle. The name of the protein is phospholambon. What is this phospholambon, sir? Told you in skeletal muscle, there is a set pump which plays a role for relaxation. This phospholambon is going to, listen very carefully, inhibit set pump. Because of this important inhibition, what is the role of phospholambon? It is going to inhibit calcium storage. That means it interferes with muscle relaxation. That's all. Simple. Serca pump is for muscle relaxation. Serca pump stores calcium. Our phospholambon is going to inhibit Serca pump, thereby inhibiting calcium storage, thereby interfering with muscle relaxation. Phospholambon in cardiac muscle inhibits Serca pump. Next, another high reason INICT question. See, this is action potential, electrical event. Now we will see the mechanical event, the muscle contraction. See, we have refractory periods, two important refractory periods, absolute refractory period, relative refractory period. We can clearly see the major part of contraction lies in absolute refractory period. What I told you, the major part of contraction lies in absolute refractory period. So if you give another stimulus here, there is no summation possible. No summation means no possibility of tetany.
Yes.
Cardiac muscle cannot be tetanized. Why?
એબ્સોલ્યુટમાં સમેશન ન થાય સ્કેલેટલ હોય કે પણ એબ્સોલ્યુટમાં સમેશન ન થાય પણ એબ્સોલ્યુટમાં કેવું છે કે સ્કેલેટલ મસલ છે ને એની અંદર એબ્સોલ્યુટ રિફ્રેક્ટિવ પીરિયડ બહુ ઓછો છે આની અંદર એબ્સોલ્યુટ રિફ્રેક્ટિવ પીરિયડ વધારે છે એટલે આમાં તો કાંઈ થવાનું જ નથી અને આમાં મેક્સિમમ હોય તો જ થાય. Yes, nice question.
Because major part of its contraction lies in absolute refractory period. It lies in absolute refractory period. So second stimulus can never lead to summation here. No summation means no tetany. Cardiac muscle cannot be
tetanized. Nice.
Right, guys. From cardiac muscle, let's move on to smooth muscle revision. Like cardiac muscle, you cannot also use smooth muscle to contract. It is not obey. It is also involuntary. Next important point. There are two important types of smooth muscle: single unit and multi-unit. They are located. I will tell you soon. Remember, single unit and multi-unit. Next point. In skeletal muscle sarcomere, we saw Z lines. In smooth muscle, there are no Z lines. The functional equivalent here is called as dense body.
Z line now
The most important point is coming here now. Asked many times before. There is no troponin. Remember, smooth muscle does not have troponin system. The calcium binding protein here is called manolin. Skeletal muscle, I told you, elasticity. What about smooth muscle? It is plasticity. Many times before asked, plasticity is a property of smooth muscle. Not only that, our smooth muscle can be kept in a state of sustained contraction with very little use of ATP. So contraction means you don't need to use a lot of ATPs. Smooth muscle can be kept in a state of sustained contraction exclusively in the GI tract for a prolonged period of time with the little use of ATP. We call that as latch bridge mechanism. Latch bridge mechanism, exclusive properties of smooth muscle. Now locations. Where will you see this smooth muscle? Single unit smooth muscle have very well developed gap junctions. This single unit smooth muscle is the most important type present in GIT. Not only that, our U structures. What are these U structures, sir? Uterus, yes. Urinary bladder, yes. Ureter. Single unit smooth muscle with gap junctions. This multi-unit smooth muscle in locations like iris, ciliary body in eye, or thyroerector muscle. These are all the locations of the multi-unit smooth muscle. Where will you see gap junctions? Single unit. In GIT, what type of smooth muscle? Single unit smooth
Single
muscle.
unit.
Now, in skeletal muscle, we have seen neuromuscular junction. What about smooth muscle, sir? Here, the neuromuscular junction is not well defined. It is exclusively regulated by autonomic nervous system. Smooth muscle, predominant regulatory control is autonomic nervous system, where you will see enlargement called varicosities, where these neurotransmitters are present. Autonomic nervous system neurotransmitter means it should be acetylcholine, norepinephrine. Smooth muscle, autonomic nervous system. The neurotransmitters are found in varicosities, acetylcholine, norepinephrine, neurotransmitter. And most importantly, look very, very carefully, guys. Here, one neuron is going to innervate multiple smooth muscle cells. What we call this arrangement as
Synapse and
synapse N passant. One neuron innervating multiple smooth muscle cells, forming multiple smooth muscle neuromuscular junctions. What we call that as synapse N passant. You will see this in smooth muscle, regulated by autonomic nervous system, neurotransmitters like acetylcholine, norepinephrine. Right, guys. Now we will revise synaptic transmission. There are two important types of synaptic transmission. What is the first one, sir? The first one is considered to be electric type of synapses. What is the meaning of electrical transmission, sir? It is exclusively mediated through gap junction. So this type of transmission is very, very fast actually. Gap junction mediated electric transmission. The second one is obviously chemical based transmission. What is the meaning of chemical based, sir? It's a long process. It takes time. Neurotransmitter release. It's a slow process. Neurotransmitter mediated chemical ones are the chemical synaptic transmission. We can clearly see here, it is this acetylcholine, for example, a neurotransmitter. It will act on its postsynaptic receptor, leading to development of certain potentials in the postsynaptic region. We call it as postsynaptic potentials. That we will understand next. In this important slide, electrical transmission is fast, mediated through gap junction. Chemical transmission is slow, mediated through neurotransmitters. A neurotransmitter acting in the postsynaptic receptor, you will definitely see in the postsynaptic region certain potentials, postsynaptic potentials. What are these postsynaptic potentials, sir? They can be excitatory or inhibitory. So correspondingly, we have excitatory postsynaptic potential or inhibitory postsynaptic potential. They can be either fast or slow. They can be either fast or slow. First, let's look into the fast ones. Fast excitatory, fast inhibitory. Now in this side, I'm writing excitatory postsynaptic potential, fast. Here I will write inhibitory postsynaptic potential, fast. Excitatory is because of influx of either sodium or calcium. Question, the ions they will ask you. Fast excitatory postsynaptic potential means either sodium have to come inside or calcium have to come inside, influx. Now what about IPSP? Fast. That means, remember, inside becomes negative, inhibition. What is this? Chloride. First and foremost, it is because of chloride influx or potassium ions moving out. What I told you, potassium is moving out, taking positive charges out, the inside becomes negative. Or one other important reason is potassium efflux or fast IPSP. What I told you, potentials can be either excitatory or inhibitory. They can be fast or slow. Fast EPSP, either sodium or calcium influx. Fast IPSP, the inside becomes negative, chloride influx or potassium efflux. So far we have discussed about the fast nature one. The same thing can be slow. That means correspondingly we have slow EPSP, slow IPSP. Exclusively one and only because of potassium. Very simple to remember. Potassium means we will always discuss only about the efflux. Very, very, very simple to remember. Whenever there is decrease, remember, whenever there is decrease in potassium efflux, that is the cause for slow EPSP. Whenever there is increase in potassium efflux, that is the cause for slow IPSP. Very simple. Remember guys, slow IPSP. IPSP means the inside becomes negative. Whenever there is enhanced potassium efflux out, potassium is moving out, the inside becomes negative. This increase in potassium efflux, the inside becomes negative. Very important phrase for slow IPSP. Decrease in potassium efflux, that means potassium ions are staying here. Potassium ions are staying here. It is positive. Positive means EPSP. That is slow. This is for slow nature. So this slow EPSP or IPSP is only because of potassium efflux. Only one again, potassium efflux. Decrease in potassium efflux, slow EPSP. Increase in potassium efflux, slow IPSP. That's all. Very important ones. Now, at this type of synapse, a special type of regulation. What is this, sir? Remember, this is our alpha motor neuron. Okay. It is going to synapse with our skeletal muscle. Okay. Now, this alpha motor neuron is going to send a small branch to this special type of regulatory neuron. Who is this guy? The name of this neuron is Rensa cell neuron. This alpha motor neuron activates a Rensa cell neuron, which in turn feed back and inhibit the same alpha motor neuron. What is this inhibition called? Feedback inhibition. It is exclusively mediated through which important cell? Rensa cell neuron. This Rensa cell neuron is inhibitory. it is going to inhibit our alpha motor neuron. Because it is inhibitory, it is going to use a very important neurotransmitter, glycine, for inhibition. Simple. An alpha motor neuron is activating a Renshaw cell. This Renshaw cell is giving feedback and inhibiting the same alpha motor neuron. Purpose, role, what is the need? This is mainly to control alpha motor neuron firing. This important regulation is needed to control alpha motor neuron firing. Alpha motor neuron activating a Renshaw cell. A Renshaw cell is feedback and inhibiting the same alpha motor neuron. Feedback inhibition. This is mainly to control alpha motor neuron firing. Feedback inhibition means Renshaw cell neuron inhibition. The neurotransmitter we placed here is glycine by the Renshaw cells. Alright guys, now finally we are going to revise about neurotransmitters. Very important names, many clinical relevance is there, so listen very carefully. The first neurotransmitter is actually acetylcholine. We discussed so many times. It's the neurotransmitter in neuromuscular junction. Not only that, you also have to remember this important nucleus. What is this, sir? Nucleus basalis of Maynard. Very important function. Acetylcholine is extremely critical for muscle contraction. Not only that, acetylcholine is the main neurotransmitter for REM on neurons, which is going to activate our REM sleep. Okay, function of acetylcholine in muscle contraction, REM on neurons. REM sleep is actually brought about by acetylcholine. This important toxin, botulinum toxin, is going to inhibit the release of acetylcholine. This point we have discussed. That will leading to flaccid paralysis. This toxin, botulinum toxin, inhibits release of acetylcholine. Two important receptors are there. For example, muscarinic. You see this in GI tract. Nicotinic, the second important type. You see this in the neuromuscular junction. So acetylcholine. Muscarinic receptors in GIT, nicotinic receptor in neuromuscular junction. Second, even more important one, dopamine. Particularly the pathways are always asked. Nigrostriatal means found in basal ganglia. Extremely important for motor movement control. Very important, dopamine. That's why in Parkinsonism you see paucity of movements. Dopamine is very important for regulating motor movements. This nigrostriatal pathway, basal ganglia. Mesolimbic, first important area, remember, ventral tegmental area, VTA, is considered to be the reward center. So all the things we do, we need some reward. Then you can perform better. Reward center is ventral tegmental area, dopamine. Second, remember, nucleus accumbens, which is very, very important for addiction. Addiction to a food, addiction to a person. All this addiction behavior, dopamine is very important. Nucleus accumbens. So ventral tegmental area for reward, nucleus accumbens for addiction. And finally, tuberoinfundibular. Through this pathway, dopamine is going to inhibit the release of prolactin from anterior pituitary. So prolactin release is inhibited by dopamine. Through which important pathway? Tuberoinfundibular pathway. Dopamine. Next is obviously our serotonin. It's a neurotransmitter from raffinucleus. It is also present in platelets. Very important. It keeps us in an awake state. Raffinucleus, awake state serotonin. Platelets, it play a role very, very important in platelet aggregation. So this raffinucleus is very important. Keeps us in awake state. Platelets, serotonin, platelet aggregation. Serotonin. The next neurotransmitter is norepinephrine. It is the major neurotransmitter from the locus ceruleus, which is going to activate mainly our reticular activating system, which keeps us in a wakeful state. This is what we call as the state of arousal. If this reticular activating system is activated, we will be awake all the time. Arousal. The neurotransmitter is norepinephrine from locus ceruleus, activating reticular activating system. Next, histamine is a very important neurotransmitter that is going to act in hypothalamus. Remember, it also keeps us in a wakeful state. That is why histamine blockers like chlorpheniramine maleate, citrazine, they induce sedation. Histamine is normally for wakeful state. If you block histamine, sleep or sedation ensures. So histamine from hypothalamus keeps us in a wakeful state. Next, very important, glutamate. For example, it is considered to be the major excitatory neurotransmitter. Very important. Glutamate is the neurotransmitter. For example, in hippocampus, most important location out of all. Subthalamic nucleus in basal ganglia. Glutamate is the major excitatory neurotransmitter. Very, very, very critical, needed for all of us, learning and memory. Glutamate, hippocampus, subthalamic nucleus, major excitatory neurotransmitter, learning and memory. Next, obviously, the major inhibitory neurotransmitter. Who is this major inhibitory, sir? GABA means always inhibitory, and this inhibition is exclusively brought about by hyperpolarization. Very, very important. GABA is a universal inhibitor. It produces all this inhibition by hyperpolarization. This important bacterial toxin, tetanospasmin, is going to inhibit the release of GABA. If you inhibit GABA, all our muscle contractions will not be properly regulated. This eventually lead to spastic paralysis. Very important. GABA produces inhibition by hyperpolarization. This bacterial toxin tetanospasmin is going to inhibit the release of GABA, thereby leading to spastic paralysis. Similar to GABA, one more important name, glycine. Remember, glycine is considered to be both excitatory and inhibitory. Recently, we have discussed a very important name, Rensa cell neuron. It's going to perform the function of inhibition here. With the help of what important neurotransmitter I told you? Glycine. Glycine can be both excitatory or inhibitory. When it is about Rensa cell inhibition, predominantly inhibitory with the help of neurotransmitter glycine. Physiology and forensic medicine. What is the antagonist of glycine? And other but our very well known strychnine. So the strychnine is a spinal poison, right? Strychnus nux vomica. The strychnine is considered to be the antagonist for glycine. What is that? Strychnine. Yes. And finally, we have gaseous neurotransmitter. Certain gases can act as neurotransmitter. First one is nitric oxide, very, very important in hippocampus like glutamate. Nitric oxide also plays a very important role in learning and memory. Nitric oxide in hippocampus for learning and memory. The most recent one in this addition is, remember, it's not that poisonous chemical, carbon monoxide. Inside the human body, we produce a very useful carbon monoxide, which can act as a neurotransmitter, this carbon monoxide, produced from heme. The source is heme. Enzymatic degradation of this heme gives rise to carbon monoxide. Remember, it is not coming from outside. Human body is producing this carbon monoxide from heme. A very important neurotransmitter functions. Remember, it is playing a role in olfaction. It is implicated in pain processing. Few examples. Question may definitely come. Carbon monoxide produced from heme in the human body. Carbon monoxide, neurotransmitter for olfaction, smell, pain processing. So neurotransmitters, very, very critical name, particularly physiology and psychiatry. So many disorders involve this neurotransmitter. So please do revise this, guys. Good luck and best wishes.
We ended this stream now. That was very nice one.